for you to read. Yeah, I'm not going to talk about this, but I'm going to talk about this one. Okay, the lecture 4, which is the safety. So safety is very, very important. Can you see the slide?
Yes, doctor.
Okay, as a user of the lab, the utmost important thing for you is your safety, yeah, and also your colleague safety. So for the general safety, the apparatus, okay, the equipment, the glassware, and all the sharp items, the chemical toxicity, the gases, the liquid nitrogen, the fire and radiation. These are all the general safety issue that may arise if you don't, deal with it properly. So for example, the operator, the trained personnel, or probably you, if you are the students, okay, the procedures are carried out properly, and direct protective clothing at the appropriate times. For ladies, you need to tie your hair if this is too long, and it's possible you need to change your clothes before you enter the lab, okay, and you need to change your shoes as well. For some very strict lab, you are not allowed to use your own shoe, but you have to change to the shoe that are provided by the lab. Okay, for the equipment, for general supervisor, it should be appointed to be in charge of all the equipment maintenance and safety. For example, the laminar flow hoods and also the autoclave. And particular risks include the generations of the toxic film. Also, the aerosol also must be contained either by the design of the equipment or by placing them in the film cupboard. So always remember, as I mentioned just now, for the chemical that will generate the toxic fumes, you need to put them properly, place them properly in the film hood. So for the glass, where you use a lot of glassware when you perform animal tissue culture with the sharp items. So most common injury handling is the broken glasses and also the syringe needle. So you use a lot of the glass pipette, and you don't actually, invite your friends for the sword fight, okay? Because it's very, very dangerous, okay? And the pasture pipettes should be discarded into the sharp bins here. So this must be a special bins for you to, to place the sharp items, or the broken glasses item. And the correct way, you must know the correct way how to put your, how to place your pipettes, okay? You don't press it with your end, so hard, because the neck may be broken, and at the end you may get cut, okay? And you'll probably spoil the equipment, because the pipette gun can cost up to 2,000 per unit. So when it's possible, the needles that are discarded, use a rigid plastic or the metal container, and do not attempt to bend or manipulate the needles. Never ever, because you don't know what are the cells that you have just cultured, okay, or by other people. Yeah, just imagine if you are culturing the pasteurized virus or other harmful bacteria, so you may get contaminated. You yourself may get infected later. Okay, and choose the correct size of the bulb or pipetting device to avoid the risk of the pipettes breaking at the neck. For the chemical toxicity, you use a lot of DMSO, which is the dimethyl sulfoxide. This is a very powerful solvent and skin penetrant. If you are accidentally spill it on your skin, you must run to the tap water and make sure that running water on your affected area for at least 10 to 15 minutes. Okay. And also the gases, most gases, for example, the carbon dioxide, the oxygen, and nitrogen used are not harmful in a small amount, but when it's come to a large amount and you don't handle them properly, then it can become dangerous. And also the gases should also be kept in a pressurized cylinder that are properly secured. So if you see the cylinder in the tissue culture lab, that you are on top of it, you should see the meter, and the meter will tell you the pressure, okay, the pressure, how much pressure there's in, okay, whether you need to change it or not, you can show by the reading on the meters on top of the cylinder. For liquid nitrogen, is extremely very, very, very dangerous. Yeah, so there are 3 major risks that are associated with the liquid nitrogens, which is the frostbite, the amphibiations, and explosions. The frostbite is especially if you put your finger right into the liquid nitrogen. When you pull it out, you just know it will be a hammers, then it can break into pieces. Yeah, this is very, very dangerous. And visualizations, that means it's very difficult to breathe, right? When you when you come into contact with the liquid nitrogen. So bear in mind. So when you handle the liquid nitrogen, you must wear proper outfit and also wear the correct types of glove, not the normal glove, but the glove that are specially designed for handling the liquid nitrogen. All right. And also fire. Fire is a very, very, very dangerous disaster in the lab. If you don't, the whole lab may burn down. It actually happens in UNIMAS, but it's not burned down the whole lab. But ironically, the lab that are burned down is our safety officer lab. Okay, so very important that you need to put your the chemicals which is flammable into the properly sealed container, right? So particularly fire risks that are used in the Bunsen burner with the flaming together with the alcohol for swabbing and also sterilization, because you have maybe 100% or 75% ethanol. So this chemical alcohol can cause fire. So you have to be very careful when you handle the Bunsen burner. Also to avoid fire, and always keep the alcohol for sterilizations or sterilizing the instruments or in a very minimum volumes in a narrow neck bottle or the flask. Okay, keeping in the distance from the Bunsen burner. For radiations, there are three main types of radiation, which is the injections, the radiations from the labo reagents, and also from the high energy sources. For example, injections, the radio label compound can be ingested by being splashed on the pan by aerosol generated by the pipetting or the. But in UNIMAS, we are very lucky because you are not allowed to use any radioisotope. Okay, radioisotope because it's very dangerous. Okay, we are not allowed to use. For example, the high energy or the beta and gamma emitters such as P32, I175, I121, and also the chromium-51. These are the high energy. Okay, so you need if you are working with this, you need a proper protection. For example, working behind a 2 millimeters thick lead shield or storing in a concentration isotope in the lid pot, and of course the prospect screen, which is 5 mm, which can be used at P32 at low concentration for the short period of time. And also the RAC X-ray machine, but we don't really use it. And of course, you need to always wear the barrier filter goggles to protect your eyes. Okay, and there's some risk when you do tissue culture lab, like low risk is a non-human and non-primate continuous cell line, and some well-characterized human continuous cell line. This is considered low risk. For the medium risk, there could be the poorly characterized mammalian cell line, because you don't know. It could be the transformed cell line or the tumor cell line or other cell line that could be very dangerous. So you really need to understand and make sure that the characteristic of the cell that you are culturing. For the high risk is a primary cell line derived from the human or primate tissue or blood, and also the cell line that endogenous pathogens, and the precise characterizations is dependent upon the pathogen, and also the cell lines used during the experimental infection where the categorizations and dependent upon the infecting agent. So this is a high risk. But of course, in our lab, because we only have the level 1 lab, we are not allowed to do a very dangerous experiment unless you have level 3 or level 4. Then you probably can do some virus work. For biohazard and cell culture, the viruses, especially the pathogenic for humans, they are ones of the most likely biohazard presented in by cell culture. Other hazard that may relate to the components of the cell culture media are other advantageous agents such as contaminating mycoplasma and also the cell products, some of which may be biologically active molecule with pharmacological immunomodulating or sensitizing properties. For example, COVID-19, it is suspected that this is escaped from one of the labs from Wuhan, but no people can actually confirm that. But you have to be very, very careful when you come into the work with the viruses. Okay, extremely, extremely careful. For generations, And also use of the modified cell, for example, the hybrid, the transformed cell line and cell containing the recombinant DNA can be also hazardous and could possibly result in the appearance of the modified or reactivated viruses, the known fusions of hybrid proteins, especially in the cross-species hybrid, and also the expressions of the virus or the cellular oncogene. The oncogene is a tumor gene. So it's extremely important when you come in, when you do the tissue culture, especially involved in this. But you are quite lucky because in our faculty, we are not allowed to do this, okay, because we do not have the proper facility for this. But when you're doing your master or PhD later, so you need to bear into, you have to keep in mind, bear in mind that this could be the potential hazard from the cell culture. Okay. And also the laboratory workers should never culture the cell derived from their own body and don't be kepochi, culture your own cell line. Okay, the in vitro transformations or the genetic modification could result in the Mendelian disease and also all the expressions of the unusual pharmacology active protein, which were to be accidentally inoculated into the journal. So never ever do that. And also human cell should be obtained from the individual having no association with the experimental work, right? And genetically modified organism, you need to handle them properly and you need the license to perform this. For example, like me, I have the license from Home Office from the UK and also the Malaysia Ethics Committee for me to do all this genetically modified, because I did some modifications, genetic modifications organism. I cloned some animal, okay, like Biosafety Act 2007 for Malaysia. So these are the license that you need to obtain before you can perform the experiment for GMO. For example, you have the laboratory disinfectant. Okay, for the disinfectant, you can use alcohol like ethanol and isopropanol. You can also use aldehydes and also the hypochlorites to disinfect your lab. Okay, but always refer to our safety officer and also a lab assistant if you wanted to disinfect. Usually, you'll be assisted by the lab assistant. Okay, so cell culture waste disposal. So tissue culture waste for the culture media, you should be inactivated for at least 2 hours in a solution of hypochlorite. It is contaminated pipette. It should be placed in the hypochlorites for 2,500 ppm, okay, parts per million overnight before you dispose by incineration. Always remember, you need to incinerate it. Of course, in Unimas, you don't incinerate because there is a company come and collect it and they will incinerate for you. For the solid waste, such as the flask, the centrifuge tubes, and contaminated gloves, tissue, it should be placed inside the heavy-duty sack for contamination well and incinerate. Usually, they will be autoclaved before they are sent for incineration. If all the possible waste should be incinerated rather than autoclaved. For the waste from the special licensed laboratory, so those handling genetic modified level 2 organism require special treatments of tracking, of course. And these are the biohazard procedure and suggests level of contaminants for your reference. And I think this is the last slide. I think that's all for today. Okay, any question, class?
No, doctor.
Are you? No? All right. If no, anyone need to scan?